Good morning, everyone. It's a real pleasure to be here. I will, as Pascal was just telling me, we have about 1.5 hours for a couple of slides. So to fill that time, it's probably good if you guys ask questions whenever, and we can stop at any point. Yes. So let's get going. So we, like many, are motivated by this book, which I'm sure that most of you are aware of. So this is, of course, on growth and form, written by Darcy Thompson, published about 100 years ago. And he, I guess, was the first to really outline the problem of morphogenesis. Okay, so of course, morphogenesis, generation of form. Here you see a few examples. And an important question to ask is, how do all these complex structures arise? Here's a worm. We'll talk more than a little bit about worms. Here's a bird and here's a fish. Okay, and of course, if we step back and go to the textbooks, there's developmental programs running. And I'll just give you two flavors of such programs. Of course, one famously put forward by Louis Volpert, the French flag model, where you have perhaps a region in a tissue that secretes a morphogen. There's a rate of degradation across the entire tissue of that morphogen. The morphogen diffuses, and diffusion together with degradation sets a length. So there you get an exponentially decaying gradient of the morphogen. The cells in the tissue then just read out absolute concentration and make cell fate decision in response to the local concentration of the gradient that they measure. Most famous example of such a type of system perhaps is the bicoid gap system in the fly. So in contrast to those types of models of developmental pattern formation, we have also this class, which is of course put forward most by Alan Turing in essentially his famous 1952 publication. So here what we have is local interactions between molecules that can diffuse and react with each other. Okay, and differences in diffusion coefficients of different classes of molecules, together with appropriate nonlinear interactions, can give rise to spontaneous pattern formation. And perhaps the most, at least my favorite example of such a system is digit specification in the mouse, which was characterized a couple of years back by James Sharp and colleagues. So, what I want to show you on this slide, which essentially illustrates about 6 hours in the life of a developing zebrafish. This is a movie that was recorded by the lab of Jan Huisken in Dresden, when he was still in Dresden, a couple of years back. What you're seeing is nuclei from different tissues labeled in different colors. And of course, what you can see here is that over the course of these 6 hours, this system is really forming all the things that essentially make the zebrafish a zebrafish. You see the heads and the eye lobes here and the backbone and so forth. And the point I really just want to make is that over the course of this 6 hours, this system moves from essentially round to non-round. Okay? And the point I want to make here is that all those models that I was just introducing on the slides before, they can't really capture that. Okay? So these types of descriptions, chemical species just talking to each other, giving rise to patterns, guiding cell fate decision, that's all not sufficient to really explain how shapes arise in development. So gene expression patterns are not enough. This is also a mechanical process. Okay? So, and you can see that, of course, by the fact that there's cells moving, there's whole tissues flowing, and so forth. Okay? So, the point I'd like to make here, and I guess this is what we're going to talk about for the entire next hour plus, is that if we step back and look at these types of systems, of course, there is a chemistry running. I'm going to refer to this really now as these types of regulatory pathways, biochemical interactions, local secretion, diffusion, biomolecular species talking to each other. I'm just going to call that chemistry, and when we look at as complex a patterning process as we've just seen, it's fair to state that in achieving the proper pattern, the proper shape, there is a communication between mechanics and chemistry. And what we'll get to over the next hour and such is that this can be really a two-way intertwined coupling. And if you think about the process of self-organization, it's not clear that it's always possible to clearly distinguish, to say that the chemistry is upstream of the mechanics or vice versa. You know, I believe there's many examples. I'm going to show you one of them where the patterning system per se forms by a kind of intertwined coupling, and the two classes, these two systems are really inseparable in the patterning process. And of course, it's also fair to state is that if we think about morphogenesis, and if we think about a theory of morphogenesis, that of course by construction must combine exactly this type of interaction. And again, this can be intertwined in essentially inseparable ways. Okay. It's also fair to say that, of course, biology couples mechanics at all scales. Okay, this starts at the molecular scale when we have, for example, molecular motors that burn ATP, that burn a chemical fuel, and the energy that's released by the ATP hydrolysis reaction provides free energy that's released can also be used to drive mechanical work. And we've been looking at this coupling again at all the scales. We've been essentially pursuing a three-tied approach. We've been really looking at the actual biological systems, and I'll tell you mostly for today about our work in the elegans. We've been pursuing a theory approach, and we'll get to that. And we also pursue a biophysics approach where we try to do biophysical measurements at the scale of cells, for example, by laser cutting experiments, at the scale of molecules, and I'm going to skip most of our work, or essentially all of our work at the molecular scale, but maybe just for completeness, where we use optical tweezer types of techniques to characterize motor activities and such. All right, so let's start at the molecular scale, and I'm just going to reference my favorite molecule, which is myosin, okay, and we'll essentially talk about actomyosin for the rest of for the rest of what I'll be talking about. Of course, actomyosin is a structure inside the cell that's important for setting cell shape and so forth, and this is comprised, for example, prominently by this myosin molecular motor. Again, myosin burns ATP, and here's my favorite movie of all times, where you can see this is ridiculous. This is high-speed AFM images. You record one such image every 7 times per second, and what you can see here is a double-headed myosin that's really walking along the actin filament. You can even see the pitch of the actin helix. This is so. I'm going to make a blunt, simple statement, which is, of course, a bit too simplistic. But if you take this molecular activity, you amplify it 1 billion times, and you regulate it and control it properly, you're going to get that zebrafish, developing the head lobes and so forth. So much of the force at the molecular scale that is generated to drive morphogenetic events comes from myosin. Okay. All right, so let's move to the scale of the cells. I've already told you, we're going to talk about actomyosin, and in the cell, this link between the chemistry and the regulation and the mechanics is really provided by this actomyosin cortical layer. This is a thin structure that separates the plasma membrane of the cell from the cytoplasm. It basically consists of mainly of actin filaments and myosin motor proteins. Myosin motors assembled in these so-called myosin minifilaments. The structure is really thin. It's about 100 nanometer thick. So you can almost think of it. Depends, of course, a bit on the size of the cell. But if you have a 10 micron plus sized cell and you have a 100 nanometer thick layer, this is really essentially a two-dimensional object. Okay? So this structure is responsible for setting cell shape, for driving active processes like cell division, and of course it couples to chemistry. So how does it couple to chemistry? And to study this, we spent the last more than 10 years now thinking about this coupling in a system that really that is perfect for studying these types of couplings, which is the developing C. elegans embryo, and in particular the one-cell stage C. elegans embryo. So you're seeing that here. Here you see C. elegans worm. The worm is about 0.5 millimeter long. It's a prominent model organism, and of course, if you go at home and go to your flower pots at home, you're quite likely to have some C. elegans in the soil. So the embryo, of course, is fertilized inside the mother. So it's too small here, but there's a couple of embryos lined up along the gonad in this mother worm. If you extract the embryos and you label the actin cytoskeleton and you generate a fancy 3D image, this is what you'll see. So the embryo has the shape of an egg, and there is a solid, rather solid-like eggshell around it that actually sets the shape of the cell. And what we're going to show you is not fancy 3D renderings, but really just cuts. And here what you see is a cut through the middle of the cell. So the cell membrane is right here where these markers of polarity are. So what we'll be talking about is polarity establishment in the C. elegans zygote. There's two classes of proteins, red and blue, anterior, posterior markers of polarity. They associate to the membrane, and this is of course also where the actomyosin cortical layer is. So now this is about 50 microns, this is about 20 microns, and so it's 50 by 20 by 20 in dimension, roughly. And the cortex really, again, is this thin layer that surrounds, that sits just underneath the plasma membrane. So in this system now, the PAR proteins, these markers of polarity, and we'll get to them a bit more detailed, they provide the chemistry, and this actomyosin cortex really provides the mechanics. And they're in tight proximity with each other. The cortex really just sits right underneath the membrane. And these markers of polarity, they have binding motifs, PIP2 type binding motifs that allow them to associate to the plasma membrane. So how do these structures now interact? PAR proteins provide the chemistry, cortex provides the mechanics. How do these structures really interact? So before we go there, let me just tell you a few more words. So this process is important in the life of the nematode worm, because this process is responsible for establishing the head to tail, the anterior posterior body axis. So the zygote starts off unpolarized with the red species of markers of polarity distributed across the entire surface. And then 6 to 8 minutes later, the system is polarized, and the posterior marker of polarity in blue here defines the posterior domain. The one-cell embryo will divide across this plane, so it'll give rise to two different daughter cells. And in fact, these two daughter cells have very distinct fates. This gives rise to the germline, and this gives rise to the entire somatic lineage of cells. Okay? So how does that switch from unpolarized to polarized work? And that's, in fact, what we'll be talking about, what we'll be focusing on mostly. But what was discovered a couple of years back, and this was really work pioneered by Ed Monroe, was that this switch from unpolarized to polarized is driven by flows of actomyosin on the surface of the egg. So now I'm showing you an image not cut through the middle, but really just a bottom view of that egg. We squish the eggs a little bit so you get a quasi 2D view of the bottom surface. What is labeled here is actin filaments in white and myosin motor proteins in green. And what I want you to take from this is that there's a period of about 6 to 8 minutes, where the surface, the actomyosin cortical layer of the cell, flows away from one tip of the egg towards the other tip of the egg. Okay? So this switch from unpolarized to polarized is driven by cortical flows, and we'll get to this at a bit more detail. But in essence, what's happening here is that the red species of PAR proteins, of markers of polarity, is dragged along with these cortical flows and leaves space, in essence, for the blue species to attach to the surface. Okay? So of course, that means that if we want to understand how polarization is driven, we now have to really understand how these types of flows are driven. And we've been looking at that for many years. And of course, a key question here now is how is cortical flow generated? What's the force balance that underlies cortical flows? What type of material are we really looking at? And one way to, of course, try to answer that is to try to get to invest in the best and biggest microscopes possible. And I'm going to show you what our best images are, and I'm just going to zoom into a region of this size. And if you go to a fancy turf microscope with structured illumination, which we had access to at Woods Hole last summer and this summer. This is what you can see. This is just about 4 frames, about 1 second, a bit under 1 second apart. And what you can see here is essentially every actin filament and every myosin minifilament, how they organize to drive these flows. So it's flowing, it's playing back and forth. Okay, so of course we're now in the process of tracking all of the minifilaments and trying to find all the actin filaments and how they're oriented and so forth. But if you just look at this type of at movies like this one, of course, what you might take from this is the impression you might get is that this is really a network, of course, and perhaps an elastic-like meshwork of interconnected actin filaments and myosin motor proteins, myosin minifilaments in this meshwork generate forces, and this somehow drives the flow. So the first thing to note is that it's, of course, elastic on short times, but the system is also characterized by a huge amount of turnover. So I'm not going to show you the data for this, but if you do a photobleaching experiment and really turn off the GFPs for the GFP molecules in a particular region, what you realize, what you observe is that the cortex really regrows very quickly. There's a huge amount of turnover. And within about 20 to 30 seconds, it's particularly quick in this system, within 20 to 30 seconds, all the actin filaments and all the myosin motor proteins are replaced by new ones that come from the cytosol. So the elastic stresses will relax over the turnover time constant. So really, this system is much more fluid-like than elastic. Okay. So if we now think about the force balance that underlies cortical flows, and if we think about what type of material this really is, this brings us to physics of active matter and active gel theory, and this is probably the one place in the world where I don't need to say that much about this, because this is essentially where this all originated. In a set of papers, from people like Jean-François Journy, Jacques Pro, Frank Jülicher when he was still here. And so a fluid-like material, so this all brings us in the realm of active gel theory, and in particular in the viscous regime. So turnover, as I just told you, really induces fluid-like properties. This is essentially a two-dimensional object if we integrate over the height. So what we're left with is essentially a two-dimensional structure that is viscous-like and that has the ability of course, what's special about this type of fluid is that it's an active fluid which has access to chemical potential and a chemical fuel, and the burning of ATP gives rise to molecular scale force generation in this material, which can generate stresses, active stresses, which can spontaneously drive flows. Okay. So we've been looking at this and we've been inspired, and it's fair to say that all I've done is really inspired by about 45 minutes in the office of Jean-François when he was still really fully located here, when I started my own group, where he was working with Guillaume. Guillaume was just finishing his PhD, and I was actually on Guillaume's thesis committee. And I was just started my group and was thinking, what should I do with of course I had written something in the proposal to get my group, but as always, you write something in the proposal and then what you do is usually not necessarily completely related. Okay, maybe I shouldn't be telling you this, but to what you propose. But anyway, sitting in Jean-François' office and realizing that they had really thought about force balance that underlies cortical flow, we started thinking, well, we've got a system here. Let's test these types of ideas and let's really try to measure flows and so forth. So in the past 10 years, we've really been trying to find the most appropriate description, physical description, for the actomyosin cortex in the context of these morphogenetic events. And so of course, the first thing we really investigated is the force balance that underlies these flows. What we were able to do is to take these theories of active matter and reduce them to the most simple picture possible, in particular to a kind of one-dimensional description. We have a material law now that drives fluid flow, 1D material law. And the thing to take from this 1D material law is that gradients, spatial differences in active tension, and local force generation by actomyosin drive fluid flows against viscous dissipation and frictional losses, and they drive fluid flows over a significant length the surface of the egg. And in particular, we can use these simple types of descriptions to quantitatively account for the flow fields that we can measure during flow. So this is due at this scale of investigation to isotropic active stress generation in the layer. So you have essentially a disordered meshwork that would like to contract isotropically. We all know that cells, when they divide, they form an actomyosin ring. And an actomyosin ring really is a structure that is built out of filaments that are aligned with respect to each other. And a cortex that has filaments that are aligned with respect to each other can undergo anisotropic contraction. The structure can generate anisotropic active stresses. And in particular, these anisotropic, the state of alignment of the local state of alignment of the actomyosin cortex, so the order parameter, which is essentially a nematic order parameter which we introduced, can couple to shear flows to help to align actin filaments. You can see that in this movie here. So this cell is about to undergo, to constrict in the middle. There's flow into this region from one side, and what I hope you can see in this small movie here is that this compressive flow gives rise to alignment of filaments, and the aligned cortex will now contract anisotropically to generate a cleavage furrow. So this can be treated, and the force balance and the dynamics can be captured by introduction of this local order parameter and coupling of this order parameter, for example, to shear flows and to anisotropic active stress generation. And thirdly, we discovered a couple of years back that the cortex can not only generate contractile types of forces, but can also generate torques. And I'll tell you a little bit more about that. But this is exemplified in this movie here, which is a particular genetic modification. We'll get to this. But what I want you to see from this is that there's rotatory flows that are driven by anisotropic active stresses, also generated by this actomyosin cortical layer. And this is particularly interesting in the context of left-right symmetry breaking. We'll get to that. Okay, but for the next half hour and plus, we want to really stick to the most simple picture and really not so much worry about details of the mechanics here, but really ask the question: how does this active layer, active fluid layer, how does it couple to the power system, to the chemistry, to establish the right pattern at the right time? Okay. So for the purpose of the mechanics, really, how we're treating the cortical layer now is an inertialess, isotropic thin film active fluid, and we've spent a lot of time convincing ourselves that this captures the force balance that drives flows to some degree. Okay. So how does this now all help with polarization? And what I'll tell you about now is the work from two fantastic postdocs in the lab, Peter Gross and Vijay. And Vijay is already off to India and has started his own group here. So an experimentalist and a theorist that have worked together. So how does this now help with polarization? But before that, any questions? My plan was to make this interactive, but maybe it's too early, huh? So anyway, just ask questions whenever, okay? All right, so how does this help with, how do these flows really help with polarization? And the simple first small or part of an answer to this problem is that these flows, I should tell you, I haven't told you, these flows, they have the typical flow speeds are microns per minute. About 10 microns, 8 to 10 microns per minute is the peak flow speed. And these flow speeds are fast enough to overcome essentially the fusion of these rather large markers of polarity. I should also tell you that there's really complexes. There's multiple proteins associated to a power complex. These flows are large enough to affect the spatial distribution of power proteins that also diffuse. So the relevant quantity to discuss in this context is known as a Peclet number, which is the ratio of a diffusive to an effective timescale. And for both species of power protein and for these types of flows, Peclet number is of order unity, which means that that flows really transport markers of polarity, and they transport them much like logs in a river. So how you have to think about the system is that the entire surface is flowing. There's in fact a cytosolic flow inside. The cortex drives flows on the surface, and I'm not showing you the images here, but this is also, of course, dragging along the entire cytosol, and there's washing machine types, flow vortices as a ring of flow vortex. The Japanese group has been studying this inside the cytosol. So everything is being dragged along, and in particular, everything that's close to this cortical layer or even attached to the membrane is going to be dragged along with these types of flows. Okay, but that's not the end of it. Okay, and we've started. So when Peter started to work on this project, there was a hint in the literature from a couple of labs that the local state of the cortex in terms of polarization, in terms of power proteins, also seems to impact on myosin levels. Now remember, myosin, again, is of course the molecular motor that generates at the molecular scale the forces that correspond to active stresses at the continuum scale. And what we could show with photobleaching experiments is that the spontaneous rate of dissociation of myosin, which I'm plotting along this axis, depends on the concentration of the red molecule. Okay, in a way that the more red molecule, the lower the spontaneous rate of dissociation. So what that means is that wherever the surface is red, there's a reduced off rate of myosin, which means there's going to be a higher steady state level of myosin, which means that there will be increased force generation, increased active stresses, and these increased active stresses will lead to convergent flows in this region. Okay, so this is important, and you guys have to understand this because everything I'm going to talk about is based on this. Yeah.
So you need a gradient of these molecules.
So you need a gradient of these molecules, as I said before. So gradients of these molecules drive flows. Haha, so exactly. So that's great. So let's get back to this one slide here. So this constitutive, this force balance equation here really tells us that gradients in active tension drive flows. Active tension is set by the concentration of myosin. So C, we call this quantity C. We'll get to this also. In our simplest view, this is really just a function of the local concentration of myosin. And what we could do in this paper here is to say that if we measure the spatial concentration field of myosin, we can relate that quantitatively to the flow field. So we can predict the flow field from the distribution of myosin. Okay, and it's exactly such that a concentration gradient of myosin will drive flows, and the flows will be in the direction of increased myosin concentration. So the question you asked, which I haven't answered, is how you even get to this distribution here. You see this here in this inverted stain. And this is, of course, now a dynamical process, and I hope that at the end of this you're going to have a satisfactory answer, which, the myosin is itself transported by the flows, and the power proteins also control myosin on and off rates. Yeah.
There's more difference in the beginning.
Exactly. So I'm going to spend much of this class to exactly answer this question. So maybe you just have to wait a little bit. Okay. But Eric.
Is it obvious what way the causality goes with this R6?
Yeah, so hold on, because that's what we're going to talk about, exactly. So that's exactly what we'll talk about in the next couple of slides. So I'm not doing a good job at answering questions. I'm just going to, yes?
What is the friction there? Where is it?
Yeah, so I was a bit quick here. So this cortical layer sits. So here's the plasma membrane. Here's this cortical layer. We have myosin motor proteins inside, myosin minifilaments inside, and if you have if you have a region with increased myosin, this will drive flows that converge upon this region, and there's a friction coefficient because this flow will generate flows of intracellular flows of this entire cytosol inside. Of course, it's very complex. Okay, so there's dissipation inside the cell, and it's very complex, and you really have to solve a full 3D, hydrodynamic problem. What we've done is we've captured all of the dissipation inside in a simple effective friction coefficient gamma. Okay, and that friction is a pair area friction coefficient. So this pair area friction coefficient and this effective two-dimensional viscosity, they together set a length. Okay, and this length has been actually put forward first by Guillaume and Jean-François. Okay.
Maybe you can say what is the initial event that breaks the symmetry?
Yeah, again, I'm just going to have to defer you because we'll talk exactly about that. So the initial, okay, well, before I defer, one more thing. Okay, so this is not spontaneous symmetry breaking. I should say at least this. Okay, thanks for pointing that out. This is not a system, this is not a Turing system where some parameter of the system changes over time and the system spontaneously breaks symmetry. This is a controlled event, okay? And I'm going to make a big statement here. Of course, there's lots of molecules in biology and there's lots of stochasticity, but if there's one process that's not left to chance, it's development, okay? Everything is perfectly controlled. You want to make the same exact worm every single time you go through. So the controlling event here is a set of signals that come from the centrosome. Okay, so the centrosome, I'm sure you all know, is this microtubule-based structure that, for example, controls cell division and chromosome organization during cytokinesis. In C. elegans, the oocyte, the egg, the unfertilized egg, doesn't have a centrosome. The centrosome comes in from the sperm, and the centrosome is a rather big structure. It hangs around after fertilization, and just when the cell prepares for its first division, the centrosome moves towards this cortex and signals something. And we'll get to exactly what it signals. But the important point here is that this is not spontaneous symmetry breaking. This is a controlling pattern formation. Okay. All right, so where was I? Was I here? So now in light of all of this, remember that now the rate of dissociation of myosin is under control of the red species, and it's such that the spontaneous rate of dissociation is decreasing with increasing concentration, which means that you're going to have more myosin in the red area. If you have more myosin in the red area, you're going to have flows converging in the red area, and the flows are going to bring in both more myosin and more red. Okay, so you really have a positive. You potentially have a positive feedback runaway situation here. So essentially, in the simple picture, the logs are actually controlling the flows of the river. Okay, and in a way that there's going to be flow of river towards where there's lots of logs. Okay, and that's going to bring in more logs. Okay, so this is really a positive feedback situation with the potential of an instability. And we'll talk about that instability in a second, but I also like to point out that this really points towards new types of physics in terms of, in terms of establishing patterns and breaking symmetry. We've been calling this mechanochemical self-organization. In this case, you have a couple of chemical species that talk to an active layer. Inhomogeneous distributions of the regulatory species can give rise to inhomogeneous force generation in the layer, which can drive flows, and the flows can redistribute the chemistry. Okay. So, I should have put up this slide. So you have regulators that talk to each other, that control active stresses in the layer. Active stress gradients drive flows, and flows can then, of course, simply by advection transport the chemistry. So we can capture this, and this is in its most simple form. So I should say that most of what I'm telling you is really a collaboration with the group of Frank Jülicher over a decade and plus. Okay, and this is the work of Justin Boyce, who was one of the first postdocs that Frank and I were sharing when I started in Dresden. So what I'm showing you here is the quantitative equation for this one-dimensional fluid. This is similar to what I've told you before. This is for the thin film inertialess isotropic active fluid. Active stresses in the layer the total stress in the layer has two contributions. One is an active stress and one is a viscous stress. And the active stress, we call that C for contractility, is a function, and this is our simple thought experiment now, is a function of the local concentration of a regulator. Okay, we call that regulator M here. So there's two contributions to the total stress, an active contribution and a passive one. This is just a viscous stress, so shear flows, gradients in velocity, shear flows generate stresses in the surface. Okay. So the important thing here now is that we make the active stress C depend on the concentration of a regulator, as I just said. This, together with force balance, which is set by friction, as I just alluded to, completes the mechanical side of things. And now to complete the entire patterning system, what we introduce is fluxes of the regulator and a one-dimensional conservation law. So we have two fluxes of the regulator. We have a diffusive flux, which is of course proportional to the spatial changes in the concentration of the regulator, and we have an effective flux, which is proportional to the velocity of the active fluid. And here's our 1D conservation law. And what happens now is that inhomogeneities in the spatial inhomogeneities in M will give rise to active stress imbalances, which will drive flows. And the flows, again, will transport the regulator because they're going to contribute to an advective flux. Okay? And this system, I haven't specified here the type of relation between active stresses and local concentration. In the simplest assumption, you make that a linear relationship, and with enough positive feedback in that relation. So if a certain amount of regulator gives rise to enough active stress in the layer, the system become spontaneously unstable. So the state of homogeneous regulator concentration becomes spontaneously unstable with respect to small spatial perturbations, and the system evolves to a steady state where there are peaks of the regulator separated by some length, and at steady state, of course, the diffusive flux of the regulator outward is at all points in space matched by an effective flux inward, which comes from the active layer. Okay. Any questions for this here? Okay, this is key. Eric.
It's obvious that it doesn't diverge, that it has some...
Yep, so for that you need a diffusion coefficient. Okay, so the diffusion of the regulator stops the system from exploding. Okay. So the key thing here is you have a system that can form a pattern, and this is really with a single chemical species now. And of course, the key thing is that you couple the single species to an active fluid layer. Okay. So now, what I'm going to talk about now is taking this type of idea and making the chemistry a bit more realistic. Because the chemistry here, again, I told you, is just a single species. But if we think about the past system, we have multiple species. We have the red and the blue and the myosin. Okay.
There is a simple way to understand what determines the length scale between the regulators.
Yeah. So that length is essentially set here by the range of mechanical communication in the layer, which is, again, simply the hydrodynamic length. Okay? And there's one good way. I already talked about the hydrodynamic length. I mean, I've done that experiment in the talk, but it's a bit messy. So if you pour down honey on a table, okay, and you take a spoon, you wait until the honey spreads out. You take a spoon, you touch the honey, you pull it up. Okay? That's going to suck in the honey. And it's not going to suck in the honey from everywhere. It's just going to suck the honey from some local region. And that length of the size of that region is sent by the interaction strength between the honey and the layer. If you put oil between the honey and the table, you're going to suck in more from larger distances. Okay, so that's exactly the physical process that sets the hydrodynamic length here. And it's really the range of the distance that mechanical stresses communicate in the layer, given that you have friction, and friction really is an external force. Okay, is that clear, roughly? Okay, and that is essentially that length in this simple system. There's just one length, which is this mechanical length. Okay, but of course now this is maybe if you now couple this to a proper Turing-like system, you have additional length scales. Okay, and of course this can give rise to more complex patterning processes. Okay, but as we go there, and before we really fully go there, I just want to make a point here, and this point we've also already touched upon. Okay, this development really is not left to chance, and what we have here is feedback structures, feedback systems that can give rise to pattern formation and structure formation, but they're really under tight control, from upstream pathways. There's molecular guiding cues. There's maybe a signal that comes from the pathway above, and that signal is spatially distributed, and that's now somehow controlling a pattern forming process by regulating feedback. or by setting boundary conditions. It can even be physical boundary conditions, okay? And I think this is, of course, particularly interesting when you move to the scale of tissues and so forth. You can provide an elastic frame or a viscous frame for some tissue scale process. Okay, so really, again, of course, Turing systems are running, and and Turing-like feedback is present, but you're not leaving the pattern-forming process to chance. At least, in particular for those early morphogenetic events, everything is tightly regulated. You want the system to have exactly the same pattern every time, and not a different pattern every time you break symmetry, like in a Turing system. Okay, so. So we've been thinking about this, and we've been thinking about this in the framework of these combined feedback structures, patterning systems that combine, mechanics and chemistry. So, we've been calling this, so what we've been investigating in the past couple of years is really how to guide these systems of mechanochemical self-organization. Okay. So how is this patterning system controlled? And I've really just told you about one type of feedback, okay? So, which is this feedback from between the flows on the surface and the distributions of these types of species, and the fact that the red species also controls the flows, okay, by controlling Myosin concentration. So how is this patterning system really controlled so that it forms the right pattern at the right point in space and time, in essence, okay? So I've told you about that. So now we have to go a bit more into the power system. There's really two feedback structures that are operating here. Okay? So there's this mechanochemical feedback that I've mentioned, that we've been talking about in the past couple of minutes. The logs control the flow of the river. That'll bring back more logs. But there's also antagonism, and this has been studied a long time by many labs. There's antagonism between the two markers of polarity, between these two power species. So these species, these are complexes made out of many molecules that attach to the surface. There's kinases in them, and you can cross these complexes can cross-phosphorylate each other. So that means that and they're diffusing, so if a red molecule happens to diffuse into the blue domain, it gets kicked off of the membrane, and it enters the cytosol. And there's a constant exchange of molecules between a cytoplasmic pool and a surface concentration field. Okay. So two feedback structures. There's this double antagonism, and this double antagonism also ensures that there's really just two domains, and there's no mix of red and blue. There's just a red domain and a blue domain. Okay. So two feedback structures: this cross-negative feedback and this mechanochemical feedback. Okay. And there's also two, and now we come to how the system is controlled and how the system is guided. And again, it's not a spontaneous symmetry breaking event. There's, as I told you, there's the centrosome that is brought in by the sperm. It sticks around and stays in close proximity to the tip. Okay, let me drink some water here. So the sperm fertilizes the egg usually at the tip. This is due to geometry of the spermatheca, which is the organ where the oocytes are fertilized. So the centrosome doesn't move very much, then starts to move towards the surface, and then signals two things. First, it just removes myosin. Okay. Now, if you remove myosin in a particular region on the surface of the cortex, the whole cortex is going to start flowing away from that point where you've removed the myosin. It's like popping a balloon in essence. Okay. So, and that, of course, triggers flows, and the flows then transport the powers, and then the whole thing starts. But that's not the only thing it does. The centrosome also loads a set of blue molecules onto the surface, locally protects a set of blue posterior power proteins, and changes them in a way that they're protected from the antagonism by the red.
I have a philosophical question.
Oh, yes.
How do you even know what is posterior? Because
Yep.
all of this is about establishing
Yep.
this.
Yeah. So that's right. So the position of the centrosome and the position of the entry point, and in fact, just the hemisphere defines the posterior. Okay.
And
So
the centrosome knows where to go.
no. So if the centrosome comes in, this is again back to the experiments from Bob Goldstein. If the centrosome comes in over here, this will be posterior by definition. And this is the process. What I'm telling you about is the process of establishing the posterior, in essence. Okay. So the sperm entry point and just the hemisphere in which the sperm enters the cell plus this whole process of self-organization help to define the anterior-posterior axis. Okay. Okay, so it's interesting. So there's, and I just want to make that point here, and we'll get back to this. There's two feedback structures, and there's two cues, two signals from the centrosome that act as guiding cues for the self-organization process. Okay, I just want to say that here. We'll get back to that a bit. So how do we now study how these guiding cues control the self-organization process? And of course, how we study this is just by trying to be as quantitative about it as possible. So this was Peter's task for the first couple of years, really just to generate a data set that captures the spatial temporal events of polarization. And to do that, we reverted to imaging, spinning disk confocal imaging, but we try to be as quantitative about it as possible. So what Peter did is really take movies in midsection embryos, segment out the surface, and then cut this open, unwrap the surface concentration field. So I'm going to show you plots. So chymographs in essence over space and time, where over space we're going to go around the top, from the anterior to the posterior, back around the bottom. As a function of time, we can determine the actual surface amounts by plotting these transverse cuts, and really, decomposing, these fields that we can measure, and really, using a kind of a model type of approach to determine the membrane-associated amount at each point across the surface. So we do that, and we're left with these line plots, where the polarized state would look like this. So the red species, we go around the top, back around the bottom. The red species is present here, and then it's absent here, and it's present here, and the blue species is at the posterior tip, which is around the center of this chymograph. Okay. We also convert fluorescence amounts to numbers of molecules just by adding, calibrated amounts of fluorophores outside of the cell, and we generate conditions where the fluorophores are supposed to emit with similar properties. Okay, so what we're left with is this spatial temporal data set. Okay, so this is actually the spatial temporal chain of events that give rise to polarization and anterior-posterior body axis establishment. So again, this is the chymograph. This is going from this is going from the anterior to the posterior. So from the anterior to the posterior, back around to the anterior as a function of time. These are the two power concentration fields. Here, these scale bars, they really convert intensity to absolute concentrations in numbers of molecules per square micron surface. We have the same for the myosin concentration field. You see that when myosin really leaves this area, there's also flows of myosin, which is a bit hard to see in this average data set, but we can measure the flow fields of the surface, on the surface by 1D PIV, and this is the actual flow field. Okay, so this is color-coded flow velocity. So the cortex flows away from this tip. So there's a positive flow velocity here and a negative flow velocity here. Okay. And again, everything is calibrated. Okay, so this is a full data set. There's about 200,000 par proteins, 150,000 red species, 200,000 blue species. They exchange between the cytosol, so total amounts are conserved. And of course, we can capture all of this. So what do the cues now do? Well, the first cue, as I told you, from the centrosome really just punches a hole, as you can see here. You can really, you can just see that here. So this punches the hole into the, removes myosin from the tip. That starts to drive flows, and then the flows transport everyone. Okay. The second cue protects whatever blue associates spontaneously to the cortex, to the membrane in the posterior region, from the antagonism by the red. So generates a species of blue that cannot be removed by the red, but that species of blue can still remove red. Okay, so this also, of course, impacts on the self-organization process. Yes?
I have a question. I just wanted to
Yeah.
Are you 100% sure that for fertilization of the sperm, it always comes from the longitudinal ends of it?
Yep. So we're not, and it doesn't. Okay. So, and this is the experiments that I was alluding to from Bob Goldstein. So if it doesn't matter where along this hemisphere the sperm enters, if somehow the system can self-organize and find the long axis of the cell. And that process we're just looking at, and this is important, this is a good point that you're raising here. So what we've done for this data set is to really just take embryos that are within, I can't remember now, X degrees, X microns away from the tip, okay? So most, I would say 80% of the embryos are really pretty much aligned already when they fertilize, and between 20 and 30% of the embryos polarize from the side. To generate a coherent data set, we've just restricted ourselves for this analysis to events where polarization happened from actually at the tip, okay?
Because from what I was thinking, if it was not restricted to the longitudinal end, and it's entered from any other side,
Yes.
and the centrosome is left there,
Yes.
then the best should let the despolarization
Yes.
go from any other side.
Yes. So.
Will not be
Which wouldn't be good, and which exactly. And what happens there? I don't have a movie here, but if you look at individual movies, let's say the centrosome were to polarize here, which would be whatever up on this side. What you would see, and we have these movies, except we've removed them from this data set, is that as the polarization proceeds, this thing moves back to the center quite quickly. Okay, and this probably has to do with a kind of curvature sensing property of the system of self-organization. And there's in principle two ways by which such a system could sense curvature, either the PAR protein, or well, there's a couple of ways. Either the PAR proteins could directly sense curvature, or their diffusion senses curvature, or the mechanics senses curvature. And that's, in fact, the latter is our favorite hypothesis at the moment, because there's two ways. There's intracellular flows driven by the surface flows that can reposition the centrosome. Okay, and this can be curvature sensitive. And there's kind of hoop stresses that can arise in the surface stress field that can also help to rotate the cell inside the eggshell.
so far that has not been incorporated into.
Yeah, so we're just doing that at the moment. Okay. Yeah.
Excuse me.
Yes.
In the middle figure at the very long time,
Yeah.
I can see that at the middle some myosins emerge.
Yeah. So I think this is a bit of an issue because what happens here is that this leaves a little bit the top surface, and so we're not totally sure if this is a bit of an artifact in our imaging, but we also see. So what does happen here is that the centrosome starts to also move away from the tip a little bit. Okay, since the centrosome also keeps removing myosin, as the centrosome moves away, maybe it comes back a little bit, okay, over time. But at our current description, doesn't really capture this. Okay.
And also, I don't know if I exactly remember that correctly, but you mentioned that the myosin is recovered very fast.
Yes.
So what is the reason that the middle region that there is no myosin
Yeah.
recovers there?
Yeah. So this brings us to the whole feedback structure. Okay, because of course this is due to the feedback structure. Okay, so there's two. So first of all, there's quite fast flows here. Okay, so if myosin were to associate here, it would also be dragged away. So at steady state, you're going to keep depleting the myosin from that region. Number one. Second, the Q is active over some time. Okay, and I'll show you the spatial temporal activity of the Q. So the Q would also just keep removing the myosin. And then the PAR system feeds back to myosin. Okay, so the PAR systems also, the red and blue species also control myosin levels. Okay, so and it's the interplay of these three effects that really gives rise to this spatial temporal pattern here. Okay, so the first Q, let me just tell you. So of course now we can try to measure as much as we can, and for the as I've told you, the red species controls the off rate of myosin, and the time constant is approximately 10 seconds. Okay, so with 10-second response time, the myosin field responds to the red field. Okay, and tries to look like the red field in essence, in simple language. So the response time here is 10 seconds. The flows essentially the flow speeds are quite fast for the microns per minute. These are about 6 to 8 minutes in total. So the flow field is essentially instantaneous with the myosin distribution. Okay, and in a viscous material, flows are set up. There's no time in the Stokes equation in essence. So this responds essentially instantaneously. So the PAR system, of course, also has time scales, but the most important one is perhaps the time it takes a species to be removed when it enters the wrong domain. Okay, so the lifetime of either the red molecule in the blue domain or the blue molecule in the red domain is sub-second. Okay, so it's 2.2 seconds removal time, 0.5 seconds removal time. So this is a very fast feedback loop. Okay, and these two together with these two Qs now somehow control the spatial temporal evolution of events. Okay. So now I'm going to skip a lot of work, okay, and just summarize it in 2 sentences. So what Peter and Vijay then did is really to isolate the two feedback structures. We can RNAi, we have the power of RNAi, RNA-mediated interference available for C. elegans. We can just RNAi myosin, then there is a cortex, has very little myosin, has no flows, and then we just have the PAR system and its Q. We can RNAi both PAR proteins, both PAR species, then we just have the myosin cortex, okay, and just a mechanical problem, okay, and self-organization of cortical flows under control of the Qs. And we can use those two systems to calibrate fully calibrate our theory, together with, of course, a huge amount of previous work from our lab and other labs, okay. But the important point is that this fully parameterizes the system, and in essence makes the wild-type combined system a prediction, okay. And I'm going to show you our prediction for the spatial temporal evolution of PAR polarization in the wild type based on calibrating in these two subsets of data. And the only thing that we changed, this took us a year to find out, the only thing that we have to adapt between these two mutant RNAi conditions and the wild type is the total amounts of PAR proteins and myosin proteins, because they seem to systematically differ. But we can measure those levels from the wild type data set, okay? So the only thing we're adjusting is the measured amounts of proteins, because they seem to be varying between mutant conditions quite systematically. So we calibrate in these two by separating these two pathways, and then we predict the full system. Of course, this is a complex, it's about, there is a significant number of unknown parameters, but diffusion coefficients are measured by FRAP, feedback, and associated parameters so all parameters can be are significantly constrained by analyzing these two conditions independently. Okay, so there's three essentially reaction, diffusion, advection equations for the three species, and then there's conserved amounts of proteins, and then there's this force balance, surface mechanics, active flow, active fluid type of description, where again this active tension that just depends is proportional to myosin concentration. Okay, and now this is the full data set. We also, this is the other thing I forgot to say. We also use this separate, divided up approach to characterize the spatial temporal action of the two Qs. Okay, so we isolate the spatial temporal dependence, identify the spatial temporal dependence of the two Qs, exactly that same process. So what you're seeing here is the first Q, which is the myosin removal Q, how it acts over space and time. This is in essence an additional off rate imposed onto myosin by the centrosome, and it interacts around the posterior domain with a certain width, and it's active for a set amount of time. So the first Q is transient, the second Q is permanent. Okay, so this generates a species of blue that is protected from antagonism by the red, and this is the spatial temporal action profile of the second Q. So with this and with the calibrated system, the wild type essentially becomes a prediction, and here's what that really looks like. The line is the prediction, and the jiggery data set is the experimental data, and you can see it captures quite well the spatial temporal evolution of the entire system. So these again are the respective PAR concentration fields on the surface around the circumference. You can see that there's a dip of myosin that drives this flow field. So this is positive flow in the positive domain, so this is divergent flow, and this gives rise to, you can see these peaks of the red. So the red species is advected away from the tip, and you get these typical overshoot peaks from advection, together with allowing, given that this concentration goes down, the blue concentration can rise. Okay. All right. So that also means that we now have essentially a full theory available, and now we can take this calibrated analysis, this calibrated description, and try to really understand how is this now controlled? What is the mechanism by which these two guides exert control on this pattern forming process? And that's what I'm going to talk about in the next couple of minutes. And for that, what I'm going to do is I'm going to show you a kind of a simplified view of the spatial temporal event of polarization. And what I'm going to show you is the trajectory in a kind of a reduced phase space, where we're going to really look at the integrated amounts of these three species on the surface. So how many red species are on the surface, how many blue species are on the surface, and how much myosin is on the surface as a function of time. Okay, and so this is the graph. So this is the number of myosin, and sorry, this is the number of red. This is the red direction, and this is the blue direction. So each arrow is 10,000 molecules. Okay, and the system starts off unpolarized, and unpolarized means that there's a lot of red species and a lot of myosin and very little blue. Okay, so the system starts here and it moves to a polarized state where both red and myosin are reduced. The myosin is of course also restricted essentially to the red, and there's also a significant amount of blue. And the way that the system reaches this point is along this type of trajectory, and again it takes between roughly 6 minutes. Okay, so this is this trajectory in this kind of reduced description, and what we can now do is we can analyze this trajectory in our calibrated theory. We can do the following thing. We can take all of the terms that I've just glanced by you. So the system, of course, has a number of feedback structures. As I told you, there's antagonism between red and blue. There's this mechanochemical feedback. And there's also terms in the theory that correspond to guidance terms, okay, where extra myosin is removed. And in particular, if you go to this reduced description, it's very easy to identify. There's a unique, clear identification of which terms in the theory correspond to self-organization, internal feedback. Oops. Okay. And which terms in the description correspond to changing surface concentration as driven by guidance. Okay. And what we're going to do is we're going to look at the time rate changes of these three quantities and how they and in particular those contributions from guidance and from self-organization. I'm going to project these time rate changes along the tangent of this curve. Okay, so this is what this looks like. Okay, so what you see is that guiding cues, which are dark blue, really pull the trajectory forward for much of the entire trajectory, and only at the very end you see that self-organization really takes over and pulls the system forward. Okay, so for most of the trajectory, guiding cues are guidance. So the system is really under control of guidance for most of the trajectory, and only, we call this guidance release, only after about 260 seconds the system switches and self-organization takes over to drive evolution of the system for the last bit. Okay, so that's one point that's worth noting. And I think it's interesting because when we started this project, I was of the clear opinion that this is really complete self-organization. What this is telling us is quite the opposite. This is quite deterministic, and everything's under tight control for most of the trajectory. Okay? So cues determine dynamics for most part, and this is really more deterministic than self-organized. The second interesting point is what we call the critical point, which is about after 60 seconds, which is if you now, in the theory, remove all cues prior to 60 seconds, the system would evolve back to the initial homogeneous unpolarized conformation. If you remove guidance after the 60 seconds, the system would evolve, is past this critical tipping point and would evolve to the pattern state. It would do so with different dynamics, but it would get there. Okay? So that's the critical point. And of course, what's nice to realize here is that the system, the guidance release point is quite far beyond the critical point. Okay, so this is in essence the transition state, and of course it makes sense. Okay, I know from my own experience, if you teach your kids how to ride the bike, you really want to make sure you don't let go too early. Okay, so this system is really letting go far beyond the transition state, probably to confer robustness. Okay, and maybe the last type of data that I'm going to show you here is we can also test the prediction. We can test whether this is really an appropriate, whether our theory properly captures the transition state, and we can do that. So here I told you in theory we just removed the guidance at different time points and asked when does it stop coming back to the initial state.
What does it mean again to remove the guidance?
So in theory we can just turn off all the terms associated with guidance. Okay. In the experiment, which is what I'm going to show you now, we can do that exact experiment. We can recapitulate that by taking a laser and just blasting the centrosome. Okay, that destroys the centrosome, and then there's no more guidance. And we now do that at different times after the process has started, and we do this under an RNAi condition where there's only one Q. Okay, so in essence, now what we're going to do is we're going to completely remove cortical flows from the equation. So there's a myosin light chain RNAi condition. So it's only the path system, and we ask, you know, so what we're going to do is we're going to ablate the centrosome with a laser, and at the time that we ablate the centrosome, we measure the extent of this domain. Okay. And we measure it, what I'm going to show you, I'm just going to show you the upper half of this, okay, not the whole distribution like usually. So this is one such condition. This is, so we're going to measure the concentration field of the blue species, which the blue species I'm now showing in green. Sorry about that. Okay, so this is the blue species. And this is the predicted transition state. Oops. Okay, so the width is set somehow by the centrosome. So for a set width, you know, you need to have this amount of part 2, this amplitude in this distribution. If you're above this amplitude of part 2 distribution, the system is going to evolve to the pattern state. If you're below that amplitude in initial par 2 concentration field, you're going to evolve back to the unpolarized state. Okay, so this is the predicted transition state. Now we ablate centrosomes in many experiments, and for each experiment we measure the extent of that domain, and this is how the data looks. So for all the yellow distributions, the system ends up polarizing. For all the gray one, the system doesn't polarize. Okay, so this matches to some degree the theory prediction for the transition state. Okay. Okay, so with that, I'm at the end. So what do we learn, at least for this part? And if you want, I can tell you a little bit more, but we can also just ask questions. So what do we learn from the system? First of all, it's not spontaneous symmetry breaking. Everything is tightly controlled. So in the language of dynamical systems theory, this really means that feedback is subcritical. There's two feedback systems running, the par cross negative feedback, the mechanochemical feedback, but none of these feedback systems are strong enough to destabilize the homogeneous state. They're waiting for a cue. Okay? And the guiding cues now drive the system well past the critical point for polarization. If you want, there's a dynamical system with two stable states, unpolarized, polarized, and if guiding cues really need, if you let go too early, the system would have there's basically two attractors here. If you let go too early, the system would come back to the unpatterned state, and what the cues really do is drive the system well past this critical point, and they then let go, and the system then just evolves to the pattern state. And I guess the key thing here is this is a mechanism of pattern formation. You know, if you impose control on a pattern forming system like this, you have a mechanism of pattern formation that avoids local instabilities. And of course, the local instabilities are exactly what you want to avoid if you want to make this deterministic. Mm-hmm. All right, and the last thing I want to say here is that really, as I just told you before, there's really two types of feedback here. There's this mechanochemical feedback, there's this double negative feedback, there's Turing-like negative feedback, and there's two guiding cues. And of course, I'm just a hypothesis. We like to think now that maybe that's a general principle. If you have multiple feedback structures in a pattern forming system, might be a good idea to control each one of them with its separate cue to robustly achieve polarization. Okay. All right. So now it's up to you. I could either tell you a little bit about talk generation. He says yes. Or we could ask questions. It's just a little bit, because I highlighted it already. Okay, so we're going to shift gears a little bit now and move to the level of cells and tissues. And now it's split back more to the mechanics. And I should say we're also trying to bring the chemistry in there too. Okay, but we're early days. Okay, so of course now cells interact. So back to mechanics at the level more of tissues. Cells interact mechanically via the cell cortex. And what I'll tell you is about the mechanism by which the embryo, the nematode worm, establishes this left-right body axis. Okay, and of course the key thing with left-right body axis establishment from the point of view of symmetries is that you're going to establish the first axis, which is the anterior-posterior axis, which is what you've heard about for the last hour. Okay, so that was the mechanism of establishing the major body axis. You then choose a second axis orthogonal to the first one, which is called the dorsal ventral axis. And then you have a choice. You could put the heart on the left side, you could put heart on the right side. Okay, and it's been, of course, we all have the heart always on the same sides. So this has, of course, been a question that also has actually fascinated physicists for long. You know, what mechanisms ensure that the heart is always on the left side? And in fact, it's not a problem. You can have humans that have a zeros inversus. If you change, if there's a process early on that apparently made the different choice, you can go to the doctor. You can be 88 years old. You go to the doctor. He looks for your heartbeat then, and he says you should be dead. And then he looks on the other side and okay, not dead. So you can be perfectly fine when this axis is reversed. Okay. So, and of course it's also interesting, I think, just a bit more from a philosophical point of view. We have essentially a bilaterally symmetric body plan on the outside, but in the inside, everything is asymmetric, left-right asymmetric. Okay? So the heart's on the left side, the gut winds up in a chiral way, and it turns out if you switch everything, it's fine. But if you switch half of the system, if your cardiovascular system is left-handed and your gut or whatever, the rest is right-handed, then you get severe problems, and of course you die at early embryogenesis. Okay, so if everything switched is fine, and this also implies that since this exists, that there are multiple points where left-right symmetries are repeatedly broken. Okay, and but before we get there, so of course to break left-right symmetry consistently, you need a chiral process. Okay, now in biology, all the molecules that are relevant are made up of polypeptide chains, and you have alpha helices. So the molecules are handed anyway. So you have to have Wolpert again. Louis Wolpert, is a famous paper predicted a so-called F molecule, which has a certain handedness, and that this handedness is somehow read out in the developmental process to give rise to consistent left-right symmetry breaking. So what I'm going to tell you is that for C. elegans, this handed process is the cortex. Okay, but before we get there, in vertebrates, the decisive event for left-right symmetry breaking is a fluid flow in a structure called ventral node, which sits essentially here at some early time point in development, and it's the molecular asymmetry of cilia. Okay, cilia, maybe you know, are these structures like hair-like structures that protrude from cells. They're made out of microtubules, and in particular microtubule doublets, and these have a handedness. Okay, and these cilia beat in a left-right asymmetric fashion. Okay, so here when you look from the top, there's a clockwise beat, and they're slightly shifted in the anterior-posterior direction. So if you have a cilium that beats shifted in the anterior-posterior direction, it's going to have an unproductive beat and a productive beat that moves fluids. Okay, and this then, so tilting cilia in the anterior-posterior direction generates left-right asymmetric morphogen flows. Okay, flows of morphogen. That's the idea. Okay, so this establishes the left-right body axis, and I'm saying here in some vertebrates. This was a big paper, 20 years ago, and then everybody thought it's the end of the story. But of course, now we've realized, and we already knew then, that there are multiple events of left-right symmetry breaking. The gut always winds up in a chiral fashion. This has nothing to do with cilia. In fact, birds and pigs do this differently. Okay, even higher metazoan organisms do this differently. So it's not quite clear how that works. And what I'm going to tell you is that at least for C. elegans, this works differently. Okay, and this is driven by torque generation in the actomyosin cortical layer. And again, from symmetry, this is allowed or possible, let's say. So now what we're going to do is we're going to think about this two-dimensional layer as being an active chiral fluid that can generate active contributions to the total to the stress tensor, and active antisymmetric contribution to the stress tensor, and you can have angular momentum exchange between the substrate and the cortical layer that give rise to rotatory movements and flows. So the cortex doesn't just contract, but also would like to rotate a little bit, always with the same handedness. Now, where does the handedness come from? We are investigating that at the moment. I have no answer, but we have ideas. And of course, actin is a helical molecule. Okay, so of course it comes back, it reverts back to molecular asymmetries. Actin is a helical molecule, and actin grows out of the surface. Okay, in essence, so the symmetries are appropriately broken to achieve such an effect. So here's the movie I showed you before. So actomyosin cortical flow in every embryo is chiral. Okay, even in the ones that I've been showing you, except the degree of handedness was so, the rotatory component to the flow field was minuscule. It's trackable and it has the same handedness in every embryo, but we can increase it with controlling myosin activity, and we can increase it to a degree where you have complete counter-rotatory flows across the entire C. elegans surface. Okay? So the conclusion here is that the cortex can generate active torques and chiral rotatory flows, where you can capture the force and torque balance. We can take the myosin concentration field and predict this chiral component to the flow field and so forth. I'm going to skip over that. So the idea is that this is the molecular, this is, no, not the molecular, but this is the chiral activity that drives left-right symmetry breaking. And again, this points to new physics in the sense that we have to think about not just force balance, but also torque balance in a fluid. And we have a fluid where there's molecular scale torque generation that can, so we've spent a lot of time with Frank to really understand force and torque balance in chiral fluids and how this can drive rotatory movements, left-right asymmetric movements. Okay, let me skip that. Okay, so in C. elegans, what happens now is that a couple of cell divisions later, so now we're moving away from the one cell division to the cell roughly 6 to 8 cell stage. There is a cell here, which is called ABA cell, doesn't really matter. This cell sits on top of another cell at the bottom. Okay, and on the top, there's just the eggshell. So there's adhesion to a cell on the bottom, and there's nothing on the top. And now when this cell divides, it's counter-rotating slightly during the division. Okay, always with the same handedness. So our idea now is that if you have a cell that's adherent to another cell and it's counter-rotating, it's turning on the spot like a bulldozer. Okay, like a bulldozer, rotates its two chains in opposite directions to turn on the spot. Okay, and this gives rise to about a 20-degree skew in the axis of division of the cell, and this then gives rise to a left-right asymmetric cell-cell contact pattern. Okay, and it's been shown about 30 years ago by Bill Wood that if you take a needle, you push inside, and you shift this in the other direction, you generate a situs inversus worm. Okay, so we could show that there's counter-rotatory flows associated with this 20-degree shift, and we can change the degree of counter-rotation in these flows, and by that we could show that this also controls the angle of this rotation. Okay, so the torque generation, myosin activity torque generation, drives this left-right asymmetric shift in cell-cell contact pattern. Okay, and then this is just the last data set. This is ongoing work by Lukáš in the group. We've now looked at all the subsequent divisions. Okay, and what I'm showing you is we can decompose movements in the cortex during cell division into a net rotation, which also happens. I'm going to talk about it, and a counter rotation. And what's crazy is that so again, I told you the first division separates the somatic from the germline lineages. The somatic lineage counter-rotates and the germline net rotates. And every division has these rotations. So the cell-cell arrangement is I'm going to say it optimistically, as we think dominated, quite significantly impacted by these rotatory movements. There's rotatory movements everywhere, and there's a germline soma difference in rotatory flows. Okay? And we can, for example, see that for all of these cells, the angle of rotation, what I was telling you, the spindle rotation angle, again, all the somatic lineage, the spindle rotation angle and the counter-rotating flow speeds are correlated, and there's essentially no counter-rotation speeds for the germline, and all the somatic cells undergo these counter-rotations and spindle rotations. Okay. Active torque generation and rotatory actomyosin flows are prevalent in the early C. elegans development, and these flows are required for proper cell-cell arrangement. That's the idea. Okay? And this is more of a hypothesis than a tested statement at the moment. Okay? So let's end here. Let me just get back to you and by summing this all up a little bit. it's maybe if there's one thing I want you to remember is that a theory of morphogenesis should combine mechanics with chemistry. We've mostly talked about a single cell, but what I want you to take, and here I'm just preaching, standing in this location, I'm just preaching to the choir. Okay, I think active matter physics, and of course combining that with regulation, provides an appropriate framework for investigating structure and pattern formation in conjunction with active mechanical processes. And with that, I'd just like to end by thanking all the fantastic people that I had the chance to work with over the past years. And what I've told you about really is mostly work from Peter and Vijay, and the talk discovery was Sundar and Sebastian. I showed you a little bit of Lucas's work. And with that, I'll stop here, and I'm happy to take some more questions if there are any.